Hi, my name is Shannon Spear, and I'm a recent PhD graduate from Notre Dame. Today, I'm going to talk to you about the controls on nitrous oxide production from incomplete denitrification in stream sediments. Now, denitrification is a facultative anaerobic process that converts nitrate through a series of intermediates to ultimately produce dinitrogen gas, or N2. This process is often considered a permanent removal mechanism for nitrate pollution in aquatic systems, as N2 gas can then diffuse out of the system into the atmosphere. But denitrification also has a dark side. Incomplete denitrification, represented by the leaky pipe analogy, releases nitrous oxide, or N2O, a powerful greenhouse gas with nearly 300 times the warming potential of carbon dioxide. Anthropogenic emissions of N2O are rapidly increasing, but natural sources of N2O cannot be forgotten as they make up about 70% of global emissions. In rivers and streams, denitrification is the primary source of N2O production. Headwater streams are especially important as they contribute up to 85% of global riverine emissions, and the Midwest US, where we're located at Notre Dame, is a hot spot due to excess fertilizer inputs from agriculture. N2O production is controlled by nitrate and oxygen concentrations, as well as carbon availability and temperature. We can also calculate N2O yields, which is the amount of N2O produced relative to total denitrification and is expressed as a percent. And our recent review identified that few studies have explored the interactions of multiple drivers of N2O production from incomplete denitrification. So here our goal was to understand how nitrate, carbon, and temperature influence both N2O production rates as well as yields. Now, based on the literature, we predicted how each driver might influence N2 production in gray, N2O production in green, and N2O yields in blue. We hypothesized that N2O production would increase with increasing nitrate concentration, but N2O yields would plateau as N2O production would increase more. For temperature, we predicted that N2O production would be highest at higher temperatures, but yields would be lower because of the greater increase in N2 production with temperature. Finally, for carbon availability, we predicted that N2O production and yields will decrease with added carbon as more carbon will promote more complete denitrification. Now, we know how we might expect these variables to influence N2O production individually, but we still don't know how they'll come together to interact. So to test these hypotheses, we collected intact sediment cores from the Big Sunflower River in Mississippi, which is an agricultural watershed where they grow corn, soy, cotton, and rice. Land use here reflects many watersheds throughout the Mississippi River Basin. We incubated a total of 48 sediment cores in a flow-through setup at the USDA National Sedimentation Lab, and then dosed cores with a gradient of nitrate concentrations that we selected based on thresholds in the literature at which N2O production or yields are expected to shift. We also included 2 DOC treatments, an ambient and an added carbon treatment plus C, to test the influence of carbon limitation. And finally, we conducted experiments at 2 different temperatures. We then passively sampled inflows and outflows of our cores for nutrients as well as dissolved gases over a 48-hour period. Now, one of the really exciting things about this study is that we employed a novel membrane inlet mass spectrometry, or MIMS method, to analyze our dissolved gases. Typical methods measure N2 and N2O separately using both MIMS for N2 and gas chromatography for N2O. This requires separate samples, which decouples the measurements and increases the potential for error. Here, we were able to use the MIMS to measure N2 and N2O simultaneously. This analysis requires you to minimize CO2 interference on the MIMS, as both carbon dioxide and nitrous oxide have the same molar mass of 44. We did this by modifying the preservation technique to add sodium hydroxide to each vial first to precipitate any CO2. Then we added zinc chloride to inhibit microbial activity that may alter the gas signature. We also used a modified cryotrap of dry ice and high purity methanol, which maintained the temperature around -78 degrees. Liquid nitrogen, which is typically used, is too cold and will not allow the nitrous oxide to pass through into the spectrometer. Once we got all of the data together, we calculated aerial fluxes of dissolved gases, which allowed us to estimate denitrification rates, N2O production rates, as well as N2O yields. So to jump into some results, let's first talk about how multiple drivers influence N2O production. I'm showing you denitrification rate here in the top panel, N2O production on the bottom panel across a nitrate gradient on the x-axis. Color represents temperature, where orange is 25C, blue is 15C, and then the shape represents the carbon treatment. Circle is ambient, triangle is plus C. Briefly, I want to touch on denitrification rates in that top panel. These were higher with increasing nitrate and carbon, as well as at warmer temperatures, which is consistent with many studies in the literature on denitrification, which is great. We're seeing exactly what we would expect. But when we look at N2O production, we see that the interaction between multiple drivers on N2O production was much more complex. I'm going to start with the drivers individually, starting with nitrate. And we found that patterns in N2O production varied across the nitrate gradient. At 15C, there was a peak at 2 milligrams per liter. At 25C, we saw that production remained low at ambient carbon, but increased to a plateau at nitrate concentrations greater than 2 milligrams per liter with added carbon. Temperature alone did not have a significant effect on N2O production, and N2O production was higher with added carbon at both temperatures, which suggests that elevated carbon does not necessarily promote more complete denitrification as we had originally hypothesized. Additionally, we found significant interactions between drivers where the effect of nitrate was dependent on both temperature and carbon. So shifting our focus to yields, we see that I'm showing you a similar figure here with N2O yields across the nitrate gradient, but now the top panel is 15C and the bottom panel is 25C. At 15C, we see that N2O yields peaked at 2 milligrams per liter of nitrate, but at 25C, yields plateaued at higher nitrate concentrations, particularly with added carbon. When we take a look at the effect of temperature, we see yields were highest at 15 degrees C. Note the difference in the y-axis range here. And in fact, the maximum at 25C, that warmer temperature, was actually 9 times lower than the maximum at 15 degrees C. And finally, with added carbon, yields were highest with added carbon at both temperatures. So starting to look at the bigger picture here, we found that total denitrification, that's N2 plus N2O, was positively correlated to the rate of removal of nitrate from the water column. Again, this is what we would expect based on what we know about denitrification in general. And we found that N2O production, which is what's critical to consider for climate change over the yield, that relative percent of N2O produced, was positively correlated with total denitrification, which suggests that conditions that enhance denitrification in general will also enhance N2O production. So what does this mean then? Well, one, high nitrate concentrations can sustain N2O production, particularly at warmer temperatures. This has important implications for agricultural areas where N2O production may remain high despite nutrient mitigation efforts that target nitrate. Additionally, as temperatures warm and rivers brown, which means that carbon availability increases, this may drive increased N2O production in rivers and streams. This could then create a positive feedback loop where climate change ends up driving increased temperature and carbon availability in streams, which then results in more N2O production. And then because N2O is a potent greenhouse gas, this could further enhance climate change. Thus, it's really critical that we consider interactions of multiple drivers when predicting changes in N2O production under future climate scenarios. And we need to focus our mitigation efforts on actions that can facilitate complete denitrification all the way to N2, because there is that benefit of nitrate removal from the water column when denitrification goes to completion. With that, I'd like to take a moment to thank the numerous members of the Tank Lab, as well as the National Sedimentation Lab team, for all of their help in preparing and executing these experiments. I'm also very grateful to the staff of the Center for Environmental Science and Technology for their assistance in both dissolved gas and DOC analysis. And then finally, I'd like to thank the Notre Dame Graduate School and support from the CEST Predoctoral Fellowship for funding this research. If you have any questions, feel free to email me. It's in the bottom corner here. Thanks.